So Jens Søndergaard is a senior scientist and is in charge of our trace metal lab and recently established also the Mercury isotope lab here in Institute of Ecoscience at Aarhus University, where we got equipment funded by the Carlsberg Foundation 3 years ago. Jens did a tremendous job in getting the machinery working during the challenging COVID period. And Jens has also been involved in monitoring mining activities in Greenland over the last many years. And now Jens has taken over the AMAP core program, where we monitor the Arctic environment as Frank Richer retired. And Jens is currently leading a Minamata project where he will present some of the interesting recent findings. So Jens, the floor is yours.
Thank you very much, Rune. I will try to share my screen.
Okay.
Well, I hope you see the slideshow in presentation mode.
Yes.
You do great. Thank you. First of all, I'd like to thank you, Rune, John, Sophie, and Simon for the invitation to participate in this webinar. It's certainly a great pleasure. And also thank you, Rune, for the nice introduction. Continuing on from Jeroen's very interesting presentation, this presentation will be on our results from the first year of Mercury isotope studies at the Aarhus University. You have done much research on Mercury in biota in the Arctic and in Greenland in particular during the last decades, with works led by Rune Dietz and Frank Richer, Christian Sonne and others. And the Mercury analysis are performed in our own laboratory, where we have different instruments for measuring total concentrations. However, in 2019, inspired by the great pioneering work on Mercury isotopes from Jeroen's labs and a few other labs worldwide working with the Mercury isotopes, we decided to apply to the Carlsberg Foundation for multi-collector ICP-MS instrument to expand our laboratory facilities. And Rune did an absolutely fantastic job as a lead applicant, and we were fortunate to achieve the funding in January 2020. Let's see if I can. Hope you can see the next slide now. Then, as Rune mentioned, the Corona lockdown came shortly after, and we received the instrument, 2.5 tons of equipment in 11 very large boxes, filling the entire workshop in the basement. However, despite the lockdown, we were able to slowly start to build up a new laboratory to house the instrument, with its many challenges and delays, especially due to Corona work and traveling restrictions at the time. In total, I think the thermal technicians probably spent a month over numerous visits to install and set up the instrument. And then finally, two years after, in April last year, we were able to run the first Mercury isotope measurements. And in that regard, I'm also grateful to Jeroen for pointing me in the right direction on the method part, which has probably saved me quite a lot of headaches. Measuring mercury isotopes using multi-collector ICP-MS is quite a complicated procedure that requires extensive sample preparation. It's more time-consuming compared to conventional total analysis for total mercury. Samples need to be acid digested, measured for total mercury concentrations, and then the solutions need to be diluted to the same concentration before measured on the mercury on the multi-collector instrument using a cold vapor inlet system. And just for the instrument time on the multi-collector with all the standards and blanks and control samples, it takes approximately 45 minutes in average to run one sample using our current method. Well, using this technique, we are able to measure precise isotope ratios for Mercury's 6 major stable isotopes and calculate delta values for mass-dependent fractionation and mass-independent fractionations, respectively. Started on the Mercury isotope research at AU. We were fortunate to get funding for a research project named the Wikimedia project, funded by the Danish EPA. And these are the results from that research project that I'll present now. It can be regarded as a pilot study, since very little is known on stable mercury isotopes in Greenland biota. And the project aims to measure mercury isotopes in a range of different key samples, types, and tissues in Greenland using samples that we already have collected. The project is focused mainly on key biota samples collected as part of the AMAP core biota program in Greenland. That is polar bear, ring seal, scorpion, and Arctic char from sites at Ecoscience on the east coast, Karnak in the northwest, and Kakatasswak in central West Greenland, and Isotopes in the south. In addition, we have analyzed a selection of food whales from Tasiilaq in the southeast, and will analyze some peat samples and some marine sediment from Sarkenberg and from Kakatasswak as well. The biota sample are all from AU's sample bank. It's a very large freezer that contains samples from decades of research projects and monitoring, including AMAP Greenland samples. The peat and marine sediment samples are from a good collaboration we have with Center for Permafrost at Copenhagen University. As I said, we are not finished with the analysis of the project. We're still lacking the peat and the sediment in a few biota samples, but so far we've run more than 500 samples through the instrument. The results are not yet published and not even fully interpreted. So some of the samples were just run a few weeks ago. So you're really getting to see some very fresh data now. Before showing the diagrams of the results, I'll just quickly show two diagrams here illustrating how both sources and processes can affect the isotope composition. The diagram to the left shows mercury in coal deposits worldwide, and the diagram to the right shows how different biogeochemical processes changes the mercury isotope signatures. These processes can happen in the environment, as Jerome explained, and for demethylation, it also occurs as part of the metabolism in mammals. So mercury isotopes makes it potentially possible to study both sources and processes in the mercury cycle, but obviously it can also be a huge challenge to separate the two depending on the sample type and trophic level, etc. The diagrams shows mass-dependent fractionation on the x-axis and mass-independent fractionations, the CAP 199 on the y-axis, and these are also the delta values I'll present in similar diagrams to make it easier to compare. To start with and form the basis of further spatial and temporal studies, we measured mercury isotopes in different tissues in Greenland, key samples, key species to best select the tissues for comparison. And here are the mercury isotopes in liver, muscle, and hair for 45 polar bears collected in 2019 to 21 in East Greenland. And as you can see, the liver is distinctively different, has a different signature in terms of mass-dependent fractionation from the muscle, which reflect demethylation, the transformation of methyl mercury to inorganic mercury in the livers. Studies of other mammals in the literature show the same trend. Muscle and hair show quite similar results for MDF or mass-dependent fractionation values, and both are known to consist of a high fraction of methyl mercury. Notably, however, the hair appears to have a slightly higher 199 value, as shown on the y-axis, than the muscle and the liver, which I currently don't have a good explanation for. We also did a similar comparison between liver and muscle tissue for 3 different tooth whales. We had 1 killer whale and several pilot whales and dolphins analyzed as part of Rune's change project. And these show the same trend as for the polar bear, and the difference between the liver and the muscle MDF signatures decreased with decreasing size of the whales, with the killer whale showing the largest difference and the dolphin showing the smallest difference. Finally, we did a comparison between liver and muscle for ring seals and for scorpions. The ring seals showed a distinct but smaller difference in mercury isotopes compared with the polar bears and the tooth whales. In contrast, however, the scorpions showed no difference in the isotope signatures between the liver and the muscle. And the same was the case for both Karnak, which is the shown here, and also for Kokatasoak. So in conclusion, muscle, as opposed to liver used in many heavy metal assessments, seem to be favorable for mercury isotope analysis in comparison between samples, because it's typically almost 100% methyl mercury, and the mercury is little affected by internal metabolic processes. For polar bears, the hair show a near similar MDF value to the muscle, and therefore it offers an alternative sample type, essentially without the need to kill the animal. As the next step, we also looked into the trophic level fractionation and compared the isotopes in the same tissue type via muscle for 3 different species: polar bear, ring seals, and scorpions, representing 3 different trophic levels, and sampled at the same location. And for the 3 sites, Karnak, Kokatasoak, and Kokodomit, these are the only sites we have samples. We do see a consistent pattern. And as you can see, the higher the trophic level, the higher the MDF value, as Jeroen also showed. And this is due to excretion of inorganic mercury and biomagnification of methyl mercury, introducing a shift in the mass-dependent fractionation value in the remaining methyl mercury. Interesting to note also, there was a tendency for the scorpions and the polar bears to have a higher CAB 199 value compared with the ring seal, which indicate that the mercury taken up by these species have been more exposed to photochemical activity. Polar bears do feed on ring seals, but also on other prey items, which is probably why the isotope signature for polar bear is so relatively scattered. So in conclusion, a direct comparison between mercury isotopes in biota needs to be species specific and preferable also tissue specific. With these conclusions in mind, we explored indications for spatial and temporal trends in Greenland key species. And these plots show polar bear mussel samples from Karnak and Ecoscience from the same period, the 1980s and 2009, respectively, with 15 individuals for each period and locations that we collected from our sample bank. And 2009 is the most recent year where we have polar bear samples, tissue samples from Karnak. And as you can see here, we see near similar MDF and MIF signatures for Karnak and Ecoscience in the northwest and in the east. So the data does not indicate any spatial differences in Mercury source characteristics between these sites and regions, which seem to be consistent with time. The same trend is seen here, and this is for ring seals and scorpions from Karnak and Ecoscience from the last years, which also indicate that a similar source characteristic between these two sites. However, patterns, samples of ring seals and scorpions from Kakatasuaq seem to reveal a different overall pattern, indicating that the West Greenland sources are different from the Northwest and the East with higher MDF values. Of course, we only compared 3 sites and regions. Obviously, more sites are needed to cover Greenland, but nevertheless, I think this indicates some interesting spatial variations. The Arctic chart from Isotopes in South Greenland revealed a very different pattern in terms of odd mass independent fractionation, delta 1 or Cap 199 than the marine species we analyzed. This is a landlocked Arctic char population not migrating to the sea. And as you can see, the Cap 199 signatures are much higher, above 6 in comparison to 0 to 2 for the marine species. And this indicates substantial photochemical demethylation in that freshwater environment. Other freshwater fish studies in the literature show the same trend, as Jeroen also showed, but it's nevertheless very pronounced here. The delta 1 or the Cap 199 and the Cap 201 signature relation displayed to the right. This can be used to get more insight into the actual processes, and the plot shows that the dominating process in that environment is aqueous photochemical demethylation rather than photochemical reduction. So this pronounced odd MIF signature would potentially enable us to trace this mercury source through the food chain, as this MIF signature will not change unless the mercury is exposed to sunlight. So this could be a way to differentiate between the marine and the freshwater mercury sources. And last but not least, we examined indications for temporal trends, and we focused on muscle tissue from polar bears and gathered the oldest and most recent samples we had from Karnak from 1989 and 2009, respectively, 15 of each. And similarly, we gathered the oldest and the most recent samples from Ecoscience spanning almost 40 years, from 1983 and until the last 3-year period. And we also added samples from 2009 to enable a direct comparison of the Karnak samples. And as you can see in the plots, and although there's some some scattering of data. There seems to be a time trend in both MDF and MIF signatures through the period. We only compared 3 periods, so more data will be needed to confirm this trend, but I think nevertheless it looks very interesting to me and worth looking more into. So in conclusion, the results indicate changes in mercury source characteristics in the northwest Greenland and East Greenland during the last 40 years, and perhaps also in environmental processes, since higher odd mass-independent fractionation, the CAP 199 value, may reflect more sunlight influence due to less sea ice coverage during the period. These change in sources could be either due to changes in atmospherically deposited mercury, or it could be a change in the distribution between the recent and the legacy mercury that can be released due to thawing of permafrost and ice. The change in MDF could also be due to change in prey items. We saw how this is very species specific. So it's really a complex interaction, which will require more studies to look into. So finally, some concluding remarks and perspectives so far. The first year of studies on mercury isotopes at AU have revealed distinct variations in isotope patterns in Greenland key species, both internally in the animals at the trophic level, spatially in Greenland, and temporarily during the last 40 years. So it all in all, it shows that mercury isotopes have the potential to decipher changes in mercury sources or source distributions, diet, and environmental conditions in the Arctic. This is a pilot study, and obviously more studies are needed to shed light on the spatial trends and the time trends in Greenland, including studies on both recent and legacy mercury. So a larger international project and collaboration would certainly be interesting and relevant to study mercury isotopes and sources on a broader scale. For example, using circumpolar key species such as the polar bear and the ring seal. And also I'd like to thank our sponsoring institutions for making this possible. And I also want to acknowledge that this is indeed is and has been a team effort with good colleagues at the AU and Copenhagen University. So I'll be happy to answer questions, and you're also very welcome to contact me by email. Thank you.